There are total 7 steps. How many? 7 steps. First is sample selection, sample homogenization, cell lysis, protein precipitation, DNA precipitation, DNA desalting and washing, DNA storage. सैंपल सिलेक्शन में आपको ध्यान रखना है सॉफ्ट एंड न्यू टिश्यू, इजी टू हैंडल, इजी टू ग्राइंड, easy to cell lysis, free from contaminations. In grinding homogenization process, sample should soft, without noise, sound and stroke. Smooth grinding, right? Once the grinding is done, sample mixed with the DNA isolation buffer solution. Generally, DNA isolation buffer solution containing 4 chemicals. First, maintain the pH, that is trace. Second, stop the enzyme, that is EDTA, disodium salt. Third, maintain ionic strength, that is generally used NaCl. Fourth, CTAB, SDS used as a breaking the walls of the tissues and cells. In case of the animal DNA, we will utilize generally SDS. In case of the plant's DNA, algae's DNA, fungal DNA, we utilize CTAB chemical, right? In making process, 5 stock solution, from that desired volume will take and put inside 1 tubes, 1 bottle, that become DNA isolation working solution. After that, protein precipitation, DNA precipitation, DNA desalting and washing, storage. That is common steps for all kinds of the sample. So common solution, protein precipitation, that is majorly that used by across the world is chloroform isoamyl alcohol. 24:1 ratio means 24 ml chloroform, 1 ml isoamyl alcohol. Both mix together and do a good job to precipitate the protein. They also a solvent. So color of the compounds, plants also they extract. Right? Chloroform is solvent. That is not dissolve in plant. Water, they dissolve in chloroform, alcohol, ethanol, methanol, right? So they also extract the color of plant sample. So you can learn plant have always 2 color. One is water soluble, another is water insoluble. Even yellow color, that is you see in the nature, have 2 colors compounds. One is a water soluble yellow color, some of the part is water insoluble. Both leaves together. Sometimes you see the leaf of 2 surfaces are different, like thumbs. There are 2 colors, 2 property. One is water soluble, another side is water insoluble. So that's major engineering you can see in the plants and many parts of the universe, right? Then storage. After that is storage. Next day we come do the electrophoresis. Electrophoresis, we take the agarose powder, boil it, cool it and put inside the gel tanks by adding ethidium bromide, and finally we load our sample with a small quantity of adding dye. That's called DNA loading dye. Once the dye is mixed, loaded, you can judge where sample is goes when you load it. Otherwise, watercolor liquid loaded in water, you can't judge it. So dye is called tracking dye. DNA sample loading dye, it's moves same direction as DNA move, so we also judge how much run, right? Finally DNA photograph is taken, and after that DNA fingerprinting start. First you see the DNA is there, means DNA isolation is done, your DNA, quality DNA. Then you go for dilution and then do the PCR work. In PCR you put 5 chemicals together. One is buffer, second is MgCl2, third is primer and DNTPs, 2 chemicals, enzyme, and that DNA you want to test. That also written in the literature, PCR cocktail. That chemical you added, put on the machine. After run, they makes a copy of the DNA, millions copy. Any sets of any sequence from any targets, you can create millions of copy within 2 to 4 hours. Understand? In that, we load into the electrophoresis again, same procedure, repeats. When we run into electrophoresis, the fragment is separated out. Again, we take the photograph that we taken today. Same way, that will be a final DNA fingerprinting record. Clear? That's record taken for the analysis work. In that, we get to identify genes, allele, pattern, profile, marker, diagnostic marker, disease diagnostic, cancer diagnostic, even to identify the viral strains. That is the basic things about the molecular biology techniques, one segment. Second segment of the biotechnology come to the cloning restriction ligations to clone the genes. If you put the enzyme in that DNA, they cut itself a right sequence, and if you put the ligase, they join them and put inside the bacteria that will express. Even RNA, Moderna vaccine, you know, Moderna vaccine is RNA vaccine. Directly, if you RNA put inside your body, they immediately make the proteins. And that protein can work as a vaccine. That was the Moderna vaccine based on the RNA sequences, DNA, gene sequences, right? So, RNA can be introduced as a vaccine because RNA is a final molecule that able to create protein. If you insert in the body, they can't do the gene work. They make small proteins, and that proteins can be work as a vaccine, and that successfully tested in corona vaccine, right? So further multiple genetic engineering chapter is start once you isolated the DNA, right? The procedure is same for all the samples. Any sample is solid, you have to grind. Any sample is not a solid, liquid, directly puts inside the tube and do the experiment. Like, thank you very much everyone. Thanks for this patience. Just raise your hand full in air. Everybody, raise your hand full in air. Thank you very much. Today we will take your feedbacks and learn what you next want.